My name is Vadim Sumbayev, and I'm a research group leader and director of research in Medway School of Pharmacy, Universities of Kent and Greenwich in the United Kingdom. In this video abstract, I'm going to present an overview of our work on behalf of my co-authors. Galectin-9 is a member of lectin family of proteins, which is crucially important for regulation of human immune responses, particularly because of its role in suppression of anticancer activities of cytotoxic T cells and natural killer cells. Recent evidence demonstrated that galectin-9 is highly expressed in a wide range of cancers, including the most aggressive malignancies. However, solid tumor cells at rest are known to secrete either very low amounts of galectin-9 or in most of the cases do not secrete it at all. On the other hand, blood plasma galectin-9 levels are often elevated in a wide range of cancers. The aims of our work were therefore to elucidate whether T cells are capable of inducing galectin-9 secretion in human cancer cells derived from solid malignant tumors. On the other hand, we wanted to investigate the integration of galectin-9 into anti-cancer immune evasion machinery by cooperation with immune checkpoint proteins and pathways. We found that T cells are capable of inducing galectin-9 secretion in human breast, renal, and brain cancer cells, and this takes place via two differential biochemical mechanisms. First one is associated with autophagy induced by interaction of cancer cell with a T cell, followed by merge of autophagosome with lysosome, where galectin-9 present on the surface of autophagosome is shed proteolytically and then secreted through lysosomal secretion pathway. On the other hand, this is a classic mechanism which we reported a couple of years ago for acute myeloid leukemia cells, where activation of calcium-dependent protein kinase C isoforms takes place, and this is followed by activation of matrix metalloproteinases that perform proteolytic shedding of galectin-9 together with a fragment of its protein carrier from the cell surface. We then found that galectin-9 cooperates with other immune checkpoint proteins, in particular V-domain immunoglobulin suppressor of T cell activation, or VISTA. VISTA, in cooperation with galectin-9, suppresses interleukin-2 production by T helpers, thus preventing activation of cytotoxic T cells. On the other hand, this machinery attenuates release of granzyme B from cytotoxic T cells and its injection into target cells, thus preventing induction of apoptosis in target cells, and this machinery also triggers T cells to release transforming growth factor beta, which suppresses granzyme B expression in cytotoxic T cells and induces differentiation of naive T cells into regulatory T cells. Finally, we confirmed our findings using mouse model with immunocompetent mice, where we confirmed that tumorigenic glioblastoma cells, human glioblastoma cells LN18, are capable of secreting galectin-9 in vivo. Furthermore, this human galectin-9 is associating with mouse CD3 positive T cells and affecting their cytotoxic potential. Based on our findings, we could suggest that we have discovered for the very first time that T lymphocytes are capable of inducing galectin-9 secretion in a variety of human cancer cells, which are derived from solid malignant tumors, and this takes place via differential mechanisms. On the other hand, galectin-9 integrates into cancer immune evasion machinery, cooperating with other immune checkpoint proteins, and we have specifically found it for VISTA. And based on our work, galectin-9 itself and the expression system which control its production could be considered as specific and highly efficient potential targets for immunotherapy of wide range of cancers.